So in this recording, what we want to start to do is look at the eukaryotic cell. It's a much more complex cell. And so what we're going to do is take a couple slides to overview the eukaryotic cell, and then we're going to delve into specifics, in this case, nucleus and ribosomes. And then we'll break down the cell and look at the different parts, each in turn. So again, eukaryotic cells tend to be much larger than prokaryotic cells. They're going to have DNA not naked in the cytoplasm, but rather contained in the nucleus, right? And that nucleus is made of membranes. So most of the organelles we talk about in a eukaryotic cell we call membrane bound. So just like we refer to a prokaryotic cell in our analogy as a studio apartment, we're going to think of our eukaryotic cell now as a mansion. And so just like in a studio apartment, when you walk in, you have everything that you need there in one room. In a mansion, you walk in, there are rooms for everything. So each room then is designed to optimize the activity in that room. You cook best in the kitchen. If you have a library in your mansion, you read best there. If you have an office, you work on your office stuff there. If you have a workout room, you work out there. Each room is designed and built such that the activity in there is as efficient as it can be. So again, these eukaryotic cells are going to be much larger than prokaryotic cells, and more compartments, more rooms, both in plant and animal cells. And it's that partitioning that's going to allow us to sequester the chemistry into specific environments where that chemistry then can be optimized. And we know that efficiency is going to be very, very important in the evolution of life. So let's look at some eukaryotic cell components. And we're going to start with this big purple thing in the middle of this particular diagram, and that is the nucleus. So the nucleus contains most of the cell's DNA, and it's oftentimes the most visible organelle. When we look at cells under a light microscope, this is the thing that you can see, and sometimes it's the only thing that you can see. And so what defines the nucleus? What's called the nuclear envelope. And it's interesting to note that this is a double membrane barrier, which we can see more clearly in this picture here. So we can see that the nucleus has two membranes, right? And inside we have what's called chromatin and a nucleolus. Now, but what I want you to note here is that you have these large, in this case purple, pore complexes. So these pores allow things to move in and out of the nucleus. Not only do they allow them, but they regulate it. Some things are excluded, some things are allowed to pass in or out. So what's doing this? Proteins. The pore complex is made up of proteins in that nuclear envelope that do stuff, right? Because proteins do stuff. What are they doing? They're regulating the movement of things in and out of the nucleus. Now, in addition, you can see this transmission electron micrograph of the nuclear lamina. And so that is a whole skeleton of protein fibers that you see just underneath the nuclear envelope that provides support and shape to the nucleus. This is going to become important when we talk about when the nucleus degrades or breaks down during cell replication. Right? So we have pores that control movement into and out of the nucleus, as well as the nuclear lamina as part of the anatomy of the nucleus. So the chromatin then is a general word we use for the DNA that's wrapped up in proteins inside the nucleus. There's a tremendous amount of DNA in each one of your cells, almost 2 meters in a human cell. And so you have to do a great packaging job to get that all into a very, very tiny space. And so we wrap it around proteins to accomplish that, and we'll discuss that in more detail later. But we're going to have our chromosomes, 46 for humans, all packed up and wrapped nice and neat in the nucleus. Now, the other thing that you might see in that nucleus is a smaller, darker circle, and that is where we have what's called the nucleolus. So the nucleolus is where we're going to synthesize ribosomal RNA, and the ribosome then is the other component that we want to address in this recording. And this is the site of protein synthesis. So the ribosomes are actually going to be made in the nucleus, and we're going to find them active either in the cytoplasm or attached to another compartment in the cell called the endoplasmic reticulum. And for now, we want to think about the ribosome just as a protein-producing factory. This is where RNA is read and proteins are synthesized. And so again, we'll see them either free in the cytoplasm or attached to the ER. We'll go into greater detail of the ribosome when we talk about protein synthesis. Now, ribosomes we find in both prokaryotic and eukaryotic cells. Are they exactly the same? No, they're not. They are both ribosomes. They are both the site of protein synthesis, but prokaryotic ribosomes and eukaryotic ribosomes are different enough that we can discriminate between the two, and also then this gives us a chance to use antibiotics, because some antibiotics will target prokaryotic ribosomes, the difference is there, yet not eukaryotic ribosomes, and that becomes a useful tool for us as we fight disease and infection.